So we're going to look at anaerobic respiration. And obviously we're going to look at the two different types of anaerobic respiration that take place. So first thing to notice that as anaerobic respiration uses glycolysis. And that's really important. So just to recap on glycolysis, we're going to start with our hexose sugar. Our hexose sugar is going to be phosphorylated by the addition of two PIs that come from the hydrolysis of two ATPs, so that we get a hexose phosphate. That hexose phosphate will then be converted to two 3-carbon compounds, which are called triose phosphate, or you will see us shorten them, as I have done before, to TP. That triose phosphate will then be oxidized, and in that process it's going to release hydrogens, which are going to be picked up, if you remember, by your NADs. So we're going to produce reduced NAD, and we're also going to use the energy from the oxidation of triose phosphate to produce 4 ATPs, leaving us with 2 pyruvates. So that bit hasn't changed, and that bit still takes place in the cytoplasm of the cell. Indeed, the whole of anaerobic respiration takes place in the cytoplasm of the cell. So the problem that organisms have is in aerobic respiration, they will take pyruvate onto the link and the Krebs in order to produce reduced NADs and reduced FADs, which will then go on to oxidative phosphorylation in order to produce ATP. However, you have to remember that in anaerobic respiration, we have no oxygen, which means that we have no final electron acceptor in oxidative phosphorylation. I'm just going to shorten that to OP. Therefore, oxidative phosphorylation will stop because the electrons can't come off the final electron carrier. So therefore, the whole process will stop. And therefore, Krebs and the link also stop. Because if the whole purpose of the Krebs and the link is to produce reduced NADs and reduced FADs, if we can't take them into oxidative phosphorylation, then we won't do Krebs and the link, because we don't want to keep producing reduced NADs, because if we keep producing them, we'll eventually run out of NADs completely. So basically, anaerobic respiration would only involve glycolysis. So we have our pyruvate from the end of glycolysis, and we have some reduced NADs that we made in glycolysis. But the cell's got a problem, because if it just kept doing glycolysis but didn't do anything with the pyruvates, it would make more and more and more reduced NADs until it eventually ran out of NAD completely, and the whole of glycolysis would grind to a halt. So what the cell needs to do is it needs to convert that pyruvate to something else. Now, in the case of animals, which is the first one we'll look at, that pyruvate is going to be reduced. It's going to be reduced to lactate or lactic acid. It's the same thing. And that reduction is going to occur by the reduced NADs being oxidized back to NAD, and the hydrogen ions going to reduce the pyruvate. Now, the advantage of that is that NAD can then go back to glycolysis so that more ATP can be made. And that's in the case of animals. In the case of plants and yeast, a slightly different process, but the same principle. Pyruvate, first of all, is decarboxylated to give a compound known as ethanol. Now, pyruvate is a 3-carbon compound. Ethanol is a 2-carbon compound, which means that we must produce CO2. And ethanol is then reduced to ethanol. And we know that reduction uses the hydrogens from the reduced NAD. So the reduced NAD is oxidized back to NAD. NAD is regenerated in order to produce the ethanol. And again, that will then go to glycolysis. So a really common question for them to ask in an exam is, why is pyruvate converted to ethanol or pyruvate or lactic acid? And when you are asked that question, the answer always follows the same format, which is this allows NAD to be regenerated, so that glycolysis can continue and ATP can still be produced. Remembering that in anaerobic respiration, you will only make the ATP that has been made in glycolysis, so that is 2 ATPs in total from the process. So in anaerobic respiration in animals, you will take your glucose, you will convert it to lactic acid and 2 ATPs. And in yeast and plants, you will take the glucose, you will convert it into ethanol, plus carbon dioxide, plus 2 ATPs. Look how inefficient that is compared to aerobic respiration, where we were making 38 ATPs. Okay, the other question they might ask is, why does anaerobic respiration only glycolysis? As I said before, you need to be specific that there is no oxygen to act as the final electron acceptor in oxidative phosphorylation. Therefore, oxidative phosphorylation stops. Move it down here, and we're going to look at how a respirometer could work in an anaerobic respiration. We came across them already in aerobic. So it's a slightly newer version than the one we were looking at before, but the principle is very similar. So we have a conical flask with a piece of capillary tubing coming out and a ruler, and you have a bubble. And we're going to put in the bottom, we're going to put some yeast. Now, if I didn't do anything else, this yeast would respire aerobically. And what would happen is, if you think of the equation of aerobic respiration, it would take in, say, 6 oxygen and will give out 6 carbon dioxide. And because I wasn't absorbing that carbon dioxide, my bubble's not going to move. So it's not going to be very helpful. So I need to make sure that my yeast is respiring anaerobically. And the way to do that is to take a layer of oil and put it over the surface, like so. That oil will stop oxygen coming in, which means that you can ensure that you have anaerobic respiration. We know that anaerobic respiration in yeast turns glucose to ethanol and carbon dioxide. So there's no oxygen involved, but it will produce carbon dioxide. That carbon dioxide will go into the capillary tubing and my bubble will move in that direction. And by looking at how far my bubble moves in a given amount of time, I can work out the volume of carbon dioxide that's being given off. And therefore, I can work out the rate of respiration. Okay, so you would measure how far your bubble has moved, say from there to there. That is all carbon dioxide. So you'd work out the volume of carbon dioxide that has been given out by your yeast in that given amount of time.